Welcome to BioVivacious. BioVivacious is a YouTube channel dedicated to clear fundamental concepts in biosciences. Today we are going to have a session on color reactions of amino acids. We know that there are several color reactions of amino acid which are specific for aromatic amino acid, for cysteine, for arginine, and also for the amino groups of amino acids. We will begin our discussion by looking at the Ninhydrin test. The Ninhydrin test, one of the most famous and highly specific test for aromatic amino acid, was discovered somewhere in 1910. It's a classic experiment carried out by Siegfried Ruhemann. Ruhemann should be remembered for the kind of contribution he has made towards the biochemical studies and building up hundreds of students in organic chemistry as well as in biochemistry.
He worked with amino acids and peptides. Therefore, his discovery of Ninhydrin reagent makes it one of the most valuable discovery in the history of biochemical reagents. It's very interesting for all of us to know that he was so dedicated to laboratory work so that he took up a living quarter in the laboratory. He can spend most of the time in research. The obituary note written by Dr. J.T. Hewitt and published in the Journal of the Chemical Society on Siegfried Ruhemann, Hewitt recollects Ruhemann as an excellent teacher. He writes, Ruhemann always seemed to be as pleased in giving the lectures as the audience was in hearing them. Within a short time, at the end of the lecture, he came around the laboratory asking each individual who had been present whether there was any further points that needed explanation. As a teacher of practical organic chemistry, Ruhemann was even more in his element than in the lecture room. What a tribute.
Ruhemann wanted his students to carry out research and publish. Over 40 such articles were published in collaboration with the students. This is an image of how amino acids reacts with Ninhydrin reagent to give the characteristics purple color.
The IUPAC name for Ninhydrin is 2,2-dihydroxyindane 1,3-dione. So this is a white solid powder. It's soluble in ethanol as well as in acetone. Now, what exactly is the kind of reaction that is being carried out by Ninhydrin? Ninhydrin is an oxidizing agent and it gets reduced in the process of reacting with amino acids. So it is a kind of an oxidative deamination reaction where amino groups as well as the carbon dioxide is removed in the process. In the process of oxidative deamination, a carbon dioxide and amine or ammonia or aldehyde, a hydrodentin is released. The structure of Ninhydrin is the reduced form by removal of this hydrogen. Now, two such molecules interact with an ammonia group which is released from the amino acid and it forms this kind of a structure. And this structure, this condensation product it's a reduced Ninhydrin is called Ruhmann's purple. Because the color it produced is a violet to purple color, so it is classically known as the Ruhmann's purple. Very sensitive test for the detection of amino acids. And this is basically a diketohydrin and it can be estimated at 570 nanometer as well. What happens when it is an amino acid? For example, let us look at if we are adding proline and ninhydrin. So the proline, which is an amino acid, it forms a bonding of this manner. So in other words, this is azomethene which does not give a blue or a purple color. This is a yellow colored substance. So this is one way by which we can differentiate an amino acid from an amino acid. Similarly, when ninhydrin reacts with the asparagine, which is having an amide nitrogen, if the color formed is neither purple nor yellow, it is a kind of an orange color. So these are some of the ways by which we can differentiate different amino acids by using ninhydrin. Having understood the basic principle of ninhydrin reaction, let us look at what are some of the applications of Ninhydrin test. The first and foremost, it is used in the forensic science, especially for the identification of fingerprints, amino acids or proteins in saliva. In such application, it has in forensic science. In the laboratory, it is used for the detection of the presence of amino acid, especially when we carry out thin layer chromatography. On the silica plates, spray the ninhydrin and heat it up at 110 degrees Celsius, and we will be able to see the migration of amino acid on TLC plate. There is a third application for the ninhydrin test. In the principle we have seen, it is releasing a carbon dioxide. So generally, if the carbon is treated with the C13, it can be used for identifying the age of collagen in bone. So therefore, by counting the radioactivity, one should be able to determine the age of bone. So this is another application of the ninhydrin test. A fourth application of ninhydrin test is discovered by a very famous scientist, Bruce Merrifield, in 1984. He won a Nobel Prize for the contribution to biosciences. He was involved in the peptide synthesis in laboratory. So look at how the peptide synthesis is carried out. So in the peptide synthesis, there is a resin. To the resin, a linker is attached. Now to this linker, you attach an amino acid. So we will be able to, when you attach an amino acid, we will be able to synthesize a long peptide in the laboratory. Now how do we protect the end of this peptide so that there are no unwanted reaction occurs? So generally, the amino acid that is added, the amino group is protected with the help of FMOC. So now, whether the right amino acid has been added or not, what is done is this will be cleaved off, FMOC is cleaved off, and they will test it with the ninhydrin reagent. If they are getting the color, then it is understood that amino acid has been added. So this test is basically called Kaiser test, and this is widely applicable in the case of synthesizing peptides in the laboratory. We should also remember that ninhydrin is a chemical that may cause allergy, IgE mediated allergy in certain individuals. In the short video, we looked at some of the historical development of ninhydrin test. We looked at the principle of ninhydrin test as well as some of the applications.